Hi. Hi, good morning, everyone. So we gave a few minutes since there were a lot of people on the line for the coffee break. So we're starting now. Uh, this is going to be the MetaboApps in GNPS2. It's going to be different from some of the workshops in this morning if you attended some of the other ones, because the idea is really a hands-on experience. So I'm going to show the examples that are already loaded in the server of those tools so that we gain some time instead of loading your own data. But the idea is that you keep asking questions, doubts, and also some suggestions you can send me later too. So we're going to divide this in roughly 4 sections. So I'm going to present each of the tools. You can follow me as I show them on the actual website of those tools. And then we're also going to have a few minutes in between for also another type of questions, more technical ones or more theoretical ones. But you can ask questions anytime, so it's going to be a little different from the other workshops, because the idea is to be really more dynamic and more interactive instead of just me presenting some slides and the workflows for those tools. So my name is Lurian. I'm a PhD student from Brazil. But the idea for this workshop actually started as a PhD visiting program that I was doing the Peter Dorstein's lab in UC San Diego, which is one of the labs that started and the foundation for most of those tools, as the GNPS is the platform that all those tools that we are going to present now are hosted. So together with me, Luís, which is also from Brazil. He's a postdoc at Professor Ricardo Silva at São Paulo University. They are also going to present some of those tools with me today. And the outline for this workshop, as some of you saw on the abstract, is the idea is I'm going to present an overview of the MetaboApps, but these are really diverse and a really long set of tools that it can be applied for many different applications in metabolomics. And then I'm going to present the drug and food readouts, the CMMC knowledge base, the FBmn-stats, and then we're going to present the ChemWalker, which is a new one that it wasn't on the abstract initially. And then we're going to talk about the mirrors on the GNPS servers, especially for this meeting, as it's going to be the first one hosted in South America, and we're going to talk a little bit more about that. So the instructions for now, as it is, the idea is to do a hands-on session, is for you to load all of those websites, or at least the ones that you plan to follow alongside me today. And as I said, you can ask questions anytime. Just raise your hand and we're going to try our best to explain this to you. You can ask in Portuguese, English, Spanish, and if any other language, we'll try to solve that. So we're going to do first the drug readouts. The second one's going to be the food readouts. The third one's going to be the CMMC dashboard. And last, the FBmn-stats. So you can load those into your notebook. And if there's any error, just give it a few minutes and try to reload that again, because sometimes that happens when you have a lot of computers on the same network trying to load those servers. So the idea for the MetaboApps, it's already published as a preprint now, and soon it's going to be published on the final version. It's bridging the complexity and accessibility in metabolomics. So ideally, these MetaboApps are built to streamline the process of working on the data that you've accessed and uploaded as an FBmn usually, as a GNPS job on your metabolomics data. So the idea behind all those apps, and some of them can be combined already on the platform, is to streamline the interpretation of complex metabolic data and making advanced analytics accessible to the wider research community. So those apps are built on a community effort. They are built on the collection of many thousands of spectral MSMS spectral data, and also the idea of some of those, especially for this workshop that I'm going to present the web version of them, is to give you a more easily access interpretation of this data instead of doing the coding or doing the streamlined process on the platform itself, that sometimes it's not easily interpretable, and it's not an easy visualization for some people. So right now the MetaboApps are comprised on these 4 sections. This I'm trying to use the scroller since the laser pointer is not going to work on this screen. So the first one is pattern filtering. For this part, we have the post-MN MySQL, when you can do searches for fragments and for some patterns on the mass spectral data. We also have the multi-step MySQL, when you can do fragmentations in ordering, or ideally for working with some specific classes of molecules when you know the patterns of this fragmentation, or if you want to discover new ones that you've been familiar with your data. The second one is the enrichment and ontologies. Those are based on reference data driven. So we have the MSMS spectra, and then we classify them as different levels of information based on the metadata that you provide on the repository. We have then the food and drug readouts that we're going to see in more detail now, that it's going to ideally to help you visualize and interpret the information from your biological samples connected to the information on the mass spectral data. We have the preprint already of those tools, but I want to highlight that they are continually being improved, and the repositories for them are continuously being enlarged. So if you check them from time to time, you're always going to see more and more information on that, and the data sets that are uploaded to them are going to be larger and larger from time to time. Then we're going to have the CMMC dashboard that we're going to discuss microbial information for these data sets. The third section is basically the statistical trend and multi-omics analysis. And for that, we have the FBmn-stats that I choose to discuss a little bit more today, because it gets an overview of many basically statistical tools that you're going to use, that most of us use for many of those metabolomics data. And then we have also the ChemProp2, that you can work on the proportionality between your fragments, and it can give you some new insights on that information. And also we have the Coromics, that you can do correlation between multi-omics data. And the fourth section is the repository scale investigation. And the main tool for that is the MAST. So it's the mass spectrometry public repository search tool. And for that, we have already some other tools that are based on the same idea, that are the microbiome MAST, the microbe MAST, and some other tools that you can search on the website. And I'm going to discuss a little bit more on how to access those tools beyond the ones that we are going to see on detail today. And then we also have the conjugated metabolome explorer, this one, which is also under development, which is a repository for information of new conjugates that are derived from mass spectral data. And also, some of you might ask if they are host-based, microbial-based, or monoculture. We're going to discuss a little bit more on that. And for the specifics, I ask you to check the paper, because there is a lot of information for all those tools. So, as continuing the overview, to access more information on the apps and how to access them, ideally you should check the GNPS2 documentation. If you type that directly on Google or whenever search browser you're using, it's going to pop up as probably the first result. But I'm going to share those slides later, so you can also click on all those links. It's already ready for you to click on them. And there are two options to use any of those apps, all the ones that I highlighted before. The first one is whenever you have a job done on GNPS or GNPS2, you can just go directly to the end of the page. So when you are on the task ID page, so when you see the status here of the job and it says the status done, so it is completed and it worked, you don't get any error, there's no missing information on that. You can just go to the end of this page, and then you're going to see on downstream analysis, you're going to see most of those apps that are already implemented directly to be worked on the web page of the job. So you can just click on them, and it's going to pop up a new window for you to work on the same task ID that you were before. And also, some of those, as the food readouts and the drug readouts, they are not implemented as a downstream analysis directly on the job page, but they are implemented as a web server. So that's one of the things that I'm going to show after now. And for that, it's almost the same thing. You just need the task ID of the job you've done, and then you apply that on the web server, and then it's gonna start working. So for that, the second option, if you don't, for either case, you need to have a job done on GNPS or GNPS2. So you need to have a task ID to work on that. But the second option is that you click on the direct link and you just use the task ID, as I'm going to show right now, and then you load that, and then the data from that job is going to be populated on the task ID of that tool that you're selecting. So for the MetaboApps, the drug readouts, which is the first one we're going to discuss today, it's a resource to empirically establish drug exposure records from untargeted metabolomics data. We have already a publication on Nature Communications for that one. And if you're following on your computer right now, so the first thing you'd like to do is type in drugreadouts.gnps.org, GNPS2.org, and then at the top of the screen, you're going to select a load example, and then it's going to populate two options. You can select either of them, but for this example, I'm going to select the first one, which is the HNRC dataset. So here is going to show a task ID for the example that we've made for this data set, but you can just type any other task ID from another job that you've performed before. One of the things that is different from these two than the other ones is that for this one, as we're talking about exposure, you need to have at least an idea of the threshold you'd like to select for these exposure records. You can change that later, but this is just for the initial visualization of the tool. And then you can also do directly, you can remove the information for blanks, blank samples, internal standards, pooled or QC samples, and even SRMs, so standard reference materials. But you can do that even if you're applying a different code for that sample. You just type in on this space here. You just type in whatever code you used for that sample. So it could be blank, it could be BL, whatever you used for that. You just type the code that it's in the name of the sample, and then use these dashes here for all the codes that you're using. And then those information from those samples is already going to be removed, and you see how they're going to show up later on the next step. I always like to remind, not only for the hands-on session, but whenever you're working with your own data set, that always click on the restart session button whenever you choose anything on your analysis. Not only on the initial step, but if you type something really different, or if you choose some parameter that is really different on the next steps, always restart the session just to make sure that you have the most updated parameter on the data that you're looking at. Because as it is an online server, everything can happen to your connection. So just to avoid any mistakes, restart the session if you select a different threshold, if you select a different code for samples, or if you do some refinement that I'm going to show right now. So this is going to be the initial summary when you click on the run analysis. For people that are following those steps on your computer, if you're having any trouble or any errors, just raise the hand and we'll try to solve that. As I said, the idea is to be interactive, as most as interactive as possible. So this is going to be the initial summary that it's going to be populated on your screen right now. So you have two options. To download. I mean, you know that you already have that data on your GNPS job, but if you want to check the input quantification table or the annotation table that it's being analyzed by the app, you can always download that on separate tables just to make sure to check anything. And the metrics based on the classification that it's based on the library. So we have, for the drug readouts, we have a really long mass spectral library of MSMS data that it's based on drugs and drugs analogs and also some potential drug metabolites. So you can also have the access for this library on the publication. So if you want just the library, just to check the spectral data and not do the whole workflow on the app, and do any other software or do directly on your job, you can also do that. And then it's going to be to classify the most present ones. So in this case, it's going to be antibiotics, antidepressants, statins, PPIs, antihistamines, antihypertensive, Alzheimer medications, antifungals, and HIV. But those are not the only ones. Those are just the highlights for the summary. But if you check the whole annotation table or the other inputs that I'm going to present, you can see everything that it's in there. The overall summary is going to show all the drug categories that are present on your sample, and you can also check the frequency that those metabolites are present on your sample, on your data set based on the total amount of samples. If you check, just going back here, if you select the top pharmacological classes, it's going to be populated on the same screen, but that's what you're going to see. So one of the options that you can change that visualization there, the bar chart, is to include or exclude drug analogs. As I said, you can check them individually if you prefer directly on the library. But you can just exclude that for now, just to make a simpler visualization. But if you're looking at specific analogs, then definitely include them and see what's been found in your samples. And you can also change the number of classes that are going to be shown on your bar chart. In this case, the standard, the default for the app is the first 20 ones, 20 pharmacological classes. This bar here that you have for any visualization on any of those tools, not only the drug readouts, it's going to be the same one. It's really interesting because from that you can already use that image from your data. You can already use that for a report, for a paper, for anything that you're working in your sample, because it allows you to take a snapshot directly from that chart. You can zoom in, zoom out, and also you can, for some of those data, if you select the last icon, which is the full screen one, this one here, for some visualizations, like the Sankey plots that I'm going to show in a bit, you can also have an interactive version of them. So you can see directly on the visualization how many samples, how many compounds are based on each of those classes. And at the end, you can also download the data, which is going to provide you a table of exactly what's been calculated for this bar chart. Any questions, any doubts, any errors so far? Okay, the second thing that's going to show on the summary information from your drug readout analysis is of course, the feature annotation table. So on that, as I said before, you can work directly on the app to do some edits and rerun the processing if you want to select or to exclude any of those annotations, or if you want to change any information on that based on your, and this is going to change just for your own data sets. It's not going to change on the app, or if you do anything else, you need to rerun that if you're using a new data set. You can also report any issues. So if you run something and the server get an error, you can report these issues, and someone from any of those labs, Peter Dorstein's lab, Daniel Petros or Ming-Chun Wang's, which are the labs that are responsible for running these apps and checking and making sure that they are working fine. Someone from their teams are going to either solve the problem or contact you to see what exactly happened. And then you can also add or remove any filter based on any of those columns on your metadata. Just keep in mind that you see in the bottom here this warning, and I'm going to highlight that for example, in this example data set, you're going to have, for this case, 0 low confidence annotations. So all the annotations, of course, as an example, we already know that they are high in confidence. And based on that, we have the parameters that the cosine score are. So we don't have, for this data set, this, as I said, as an example, all the cosine scores of this MS spectra are higher than 0.9, so we have 90% match. We have at least 2 matched peaks, and if you have anything different from that, or in this case lower than that, it's going to be highlighted in red on the table. In this case, we have 0 low annotation. Based on that, you can always change those settings. Those are the default ones. It's not always that you can get all the mass spectra data to be 90% confident, so be aware of that. And also, based on how strict you are, you can also change for a higher number of matched peaks based on what exactly you want to see on that. And as you change that information, as I said, you can do the restart session on the initial page, or in this case specifically, you have the option just to rerun the analysis based on the new parameters that you selected. And also keep in mind that in this case, for example, the example one, those features on this lower table that you can see just below the initial table, the annotation table, those are the excluded features. So keep in mind, especially for the ones that are directly working with exposomics or any type of exposure data, keep in mind that some things that you might not see on your annotation table are based on the default exclusion list. So always keep that in mind, that some compounds that you may be expecting to see in all of your samples are just being excluded because those are the default for the app. So always make sure on that. So for example, in this case, as we're going to talk a little bit more about food, and for the food readouts in a bit, which is our next tool, if you're looking for piperine or capsaicin based on some food, especially some peppers, you're not going to see any of those because that's just the default of the app to not show them as related to any drug or exposure annotations. So, and also you can change that on the tables. As I said, you can download each of those tables when you just scroll above those tables. The app is going to show you the option to download those tables. But just also keep in mind that, for example, for the default, the features that are being excluded are the ones based on background or in this case noise or blank samples, as low confidence, which is one of the categories we have for some of those annotations, and endogenous or food related compounds. Those are the standard ones. So if you're working with something that you already know that belongs on that categories, you can already change that just before starting your analysis. So the next part is going to be the drug detection. So for the drug detection, it's going to be already populated with the main categories, as I said before. In this case, it's going to be those ones here, which are the ones that are shown on the summary statistics. And also you have the detection comparison, which is one of the things that is highlighted on this tool, and that we also show some applications on the paper published based on that, which is the idea of keeping track of whether the parent drugs are present on your samples, or the analogs or the metabolites. And for many of those data sets and many of the works that are been doing using those tools, at least for now, these can give you many hints on the type of data you have, especially depending on the matrix you have. It can give you some ideas on whether the method that you're working on is not maybe ionization, some of those compounds, or maybe the compounds that you're searching, if you're working, for example, with brain samples, some of the compounds are not crossing the blood-brain barrier and some are, and that might be one of the reasons that you're seeing some metabolites or some analogs and maybe not the parent drug, or the other way around. And as I said before, you're always going to have on the charts, the bar charts or the Sankey plots, you're always going to have the options to expand and to download these charts. And also we have the co-occurrence analysis, which is as the default, it's also not showing, it's excluding the analogs, not showing them. We have, for this data set, we have 4 top classes for the upset plot. And in this case, we are showing all the samples, in this case, just a small data set because it's easier for the server to populate as we are doing all these steps right now. And And as you can see, so for these upset, we have the co-occurrence of the HIV protease inhibitors, the histamine 1 inhibitors, the strand transfer inhibitor, and anticholinergic drugs. And then you have the analogs are excluded for this one, and you have the number of samples that those are present. And as I said before, you can already download these as an SVG type of picture of image, so that you can already change colors or anything in any editor that you're used to work with. Also, on the bottom of some of these more detailed visualizations, like for example the upset plot, if you click here on the bottom, it gives you some hints at least. I mean, of course, it's not a full analysis and description of your data set, but it gives you some hints on how to interpret the axes and the correlations on the upset plot. And the last one is going to be the pharmacological classification overview. So as I said, for the Sankey plots, you also have an interactive version of that. But just an example, these ones going to be, for the default, we have the top 5 therapeutic areas, but you can change that for any classifier that you have on your metadata. And then you also have the pharmacological classes. In this case, we have the top 10. And then you can track which of the compounds are related to any type of therapeutic area, any type of use. And also you can use on these one, you can also change you can also check the different type of classes, as I mentioned before, if it's already known to be a food metabolite, an endogenous, a cosmetic, a personal hygiene product, and things like that. We already have that information for the drugs that are present on the library. And also you can generate the SVG file to edit something, some information if you want to change the classification that you're using for that, or just to make the image a little bit prettier. Like for example, in this case, the classification is so large that it overlapped the other tag. And for that part, for the drug readouts, I'll give some minutes before I change for the next one. And if you have questions, doubts, if you don't get anything, just hit me up. Just raise your hand and we'll try to discuss and see what's not working fine. Okay, so Professor Ricardo asked me about what to see on the actual library. So what's on the spectral database? You can check on the, so if you go directly to the web page of the information, and then you go down to how it works, it's basically gonna show up in a more summarized version of what I just presented now. But if you click on the first one, which is the Zenodo link, you're going to have access to all the libraries, so the drug analogs, the drug metabolites, and basically the parent compounds of all of these spectral libraries that are collected and that are being used to process your data on the tool. I mean, I can just show right now, of course, I cannot present all the spectra, but if you go down there, you can see at least how it's classified. So you have already, right now you have 4 versions. The version 4 is the one that it's been used for the last version of the publication. So the paper that it's that it's been published on Nature Communications. It's going to be related to version 4. And then you see that you have the two MS reference libraries, which is, in this case, the first one is the drug library spectra of drugs and metabolites, and the second one is only for the drug analogs. If you want, for any reason, just work with one of those and not use the app, you can just download the library here, and then you can select this library on your GNPS job and just work directly with one of those libraries. And also we have the files for the metadata of that information, so the pharmacological classes, the therapeutical use, annotations, and the information provided from that spectra. It's already on the metadata file. So basically you just select if you want to just use one of those, or if you want to combine with your own internal library when you're doing the job, you can just add those libraries to your job and then run it. And it's basically gonna pop up as if you open it, it's basically gonna show an CSV file with the identification of in this case for the analogs, the name that's gonna show there, so the parent compound that it's related and the difference on the delta mass, and also the parent drug identification for you to do any correlations based on the annotation itself. So, and it's going to be similar for all the other tools. Just for the drug library specifically, the curation of this MS spectra is more straightforward. So you already have all this information of the tools and the compounds that are present. So, yeah.
Maybe you explain this information somewhere.
Yeah, sure.
My question was that what kind of raw data are you kind of putting there besides the library?
Okay.
Because is it the MS data, and which format is it? Is it compatible with this one, or do we have to put it in one software, then from that software, then we generate that information and we put it there?
Okay.
And then you showed something like features and libraries. So how do we sure that these are matched with our own samples?
Okay.
And is it urine samples or any of those kind of samples?
Okay, that's a good question. I'm not sure if everyone listened. So basically, I'm sorry, what's her name?
Laxon?
Okay. Latson basically asked me how exactly the workflow works to compare your data to the drug library. If I'm correct, that's basically it, and how it's been done, the curation for that. So it's the curation of the MSMS spectral database. It's a little bit different for some of those tools. For the drug readouts, we have the information from the analysis of those drugs and the samples that we already knew that has those drugs present on them. And the idea for you to check that is, you can do that basically just looking at the visualizations. But if you really want to see the patterns and the fragmentation, if I get it right, that's part of your question. To check that, then you need to download those tables here. And then if you are not sure about some information of that, you can always check the number of peaks, as I said, the cosine score, which in this case is the standard calculation for the comparison, is the cosine score, which is the one that is also present on the GNPS job. That's why it's the same for them to work as a streamline tool. But if you are not sure, or if you want to check anything in particular, you have basically two options. One is use the libraries directly on the GNPS job, and then you can check directly on the tool itself. I'm not sure. I can show you later, but I don't have it right now. But basically, you can just open up your job and look at the annotation list, which is a standard feature for any GNPS job, and just check on the library how's the cosine for your spectra, because then you can look at both of them in another tool, which is the GNPS dashboard. So basically, you just use the USI, which is the identification for your specific spectra on the job. And if you use the USI for that, it's going to show what's the annotation that it's been populated on your sample or on the specific spectra that you're looking at. Did you get it? No. You can ask again.
Maybe we can talk later.
We have time for that. And as I said, one question probably is going to answer for the other tools too. That's why I'm saying you can, if I'm not clear enough, you can ask them, because it's going to be probably a similar question for the other tools too.
Yeah, because I got some information from what you have said, but also my point was, okay, fine. Which kind of like platform, because we have to generate our, for instance, if I go in the lab, I run maybe urine samples. I know that in urine samples, there are so many metabolites there and also drugs there. And then how do I process that data to use this GNPS2? Because that's what I want to know. I want to see how usefulness it is to my workflow.
Okay.
Is it doable in that case?
Okay. Yeah, for the, let me try to open it. Hmm. Okay, yeah. Yeah. Okay, yeah, I'll try to simplify that. So, yeah, so discussing, yeah, thank you, Professor. So, basically, your question is more related, that's what I was trying to show here, but I'll need some time to do that. But basically, for discussing the MetaboApps, we assume that you're already a user of GNPS2. I'm not trying to be rude, it's just because then I need another workshop just to explain the idea behind the GNPS platform. But just to answer your question, basically you're going to need MSMS data like regarding different instruments. It's not an issue. You just need to have high resolution MS2 data, and then you need to convert that for MZML. And then if you have that and the MGF file, and then you create an MGF file of that spectra, which you can get from MZMine, sorry, an MZMine work that you do on your samples. Basically, that's the output you're going to get. Of course, you can use other softwares, but just an idea as it is one of the most popular ones. And then you can basically add that information to the GNPS job, and then add the libraries, which the drug readouts is just part of one of those libraries that we've curated for drugs, but you're going to have a lot of them, and one of the newest ones is the conjugated library. As I said, it's still under development. But then what you can do and then when you put when you upload all that information for doing a GNPS2 job, what basically you're going to get is an information of the annotation based on which libraries you selected for that. So the drug readouts is just a way to see the results for the drug libraries. So it's a way to simplify the results that you're getting if you use, for example, the drug library. The drug libraries. So So that's one of the things. And also, and based on that, if I go back to this part of the presentation, you're going to select some, and to start this job, you're going to select some information based on how similar or on how strict you want the correlation to be based on the fragmentation of your spectra. That's one of the things behind many other parameters that you're going to select to run a GNPS job. That's the first thing. When the job is done, as I showed here, you're going to get a task ID from the platform. So basically, for that data set, for that specific parameters you selected, that's going to be the identification for you to use in any of those tools that we're going to discuss right now as the first one being the drug readouts. And then, when you have that done, one of the things is to work on the network. So ideally, when you're using, most of us are using FBmn jobs, so feature-based molecular networks, and that's one of the one of the foundations for the creation of the GNPS platform, is to look at the networks to see how the fragmentation spectra of your data is connected between analytes, between different samples, and how you can gather information from that to understand more about the compounds and the information you're getting, the chemical information you're getting from that sample. So that's the foundation, basically, in a very, very summarized way, that's the foundation of GNPS. And then, for that, you can use the downstream analysis, which you have over, I think, more than 10 different tools. So basically, you have more than 10 different types of analysis that you can do beyond that molecular network and the annotation, and to see more information on that. So one of those is the drug readouts. As I mentioned, the readouts, so the drug readouts and the food readouts, those are specifically web-based tools. But you can also use the spectral library and run a job to just see that information if you don't want to use the web-based, but just work on your data with coding and working in tables, basically. So just to keep moving on, but we can discuss that later.
Question? A very quick question.
Yeah, sure.
Yeah. One is that in the last thing that you showed, there is something called analogs excluded in the diagram. Yeah, right here. So what do you mean by that? What are the analogs? What exactly mean by that?
Okay, so the drug readouts have two separate libraries. One of the libraries, and you can access them combined or separately. So in this case, the analogs are information of the structural information of the structure of some compounds that they have minor modifications, but they are not exactly the parent drugs. So basically, you're going to have a parent drug like, let's take one here, for example. Okay, yeah, I can show on this one. So, for example, you have metoprolol, for example, a hypertension medication, but you can have slightly small modifications on that structure that are based, so the core is ideally the same, but you have, for example, I don't know, maybe a hydroxylation or a methyl group instead of an ethyl group and things like that. Those are considered analogs. And of course, you can check, as I mentioned, if you really want to go into detail, you can open up the spectra on the library and see what exactly you have for any type of drugs. But basically, you're going to see some difference on the structure that are really similar to the parent compounds, so the parent drugs, and the list is really long. But they are not also, or at least yet, not known as human metabolites of those drugs. So basically, you're going to see a similar metabolite, but ideally, we don't have information yet. Maybe they're going to move from analogs to being actual metabolites of those parent drugs. And that's basically the difference.
So do you mean that all similar-looking metoprolol and its analogs, methylated, hydroxylated, fluorinated, whatever,
yeah,
they are all clustered together as metoprolol?
No. So in this case, that's the difference. So in this case, as they are excluded, they are not being considered as metoprolol. But if you include that, then you have that information. Then you have that information here. So you see that in these one, for example, you have parent drugs only and the analogs. So that's the amount of the frequency of detection on samples that you have for, in this case, for the classes, but you're going to have that for the specific drugs as you check on the table or if you change the parameters for that visualization. Okay, so the second one, we're not going to dive into really detail on that one because they operate really similarly. But the idea for the food readouts is to learn more about the molecular fingerprints of foods, but the idea is not to only look at metabolites that are derived from food, but to see the correlations of that for the dietary intake. So basically, you can do some estimation. So this is going to be the homepage when you enter the tool. And so the idea for these one is not only to look at metabolites that are correlated from like to any type of food intake, but the idea is to do an estimation on types of food or even some specific ones depending on how strict that metabolite is for a specific food. But you can estimate some dietary intake for some types of classes. And when I show the picture, it's going to be a little bit clearer. But just talking about the workflow itself, for the food readouts, you also need to have a task ID for the job you performed on GNPS. But the difference in this case is that you also need to upload the quantification table as we're working with estimation. So in this case, you also need the quantification table as a separate resource that you need to upload for the tool. Unless you're doing the downstream analysis, that's a different thing. But you need to upload that and to upload the metadata for that samples as we are doing some more detailed correlation on that type of information and not looking just at annotation. So that's one of the things that is really different from the drug readouts. For the food readouts, is to do that estimation, you need more information on the types of samples. One, of course, is as I'm going to show at the example, is to look at the difference, for example, in diet, if you're doing a diet study. So the first information you're going to see, so one of the things is you're going to get on the food readouts is that you have different level of the biomarkers. So basically you have different ontology levels for the food that for the food information that you're looking at. And of course, as specific as you goes, it might get trickier because then you might get to a level that you can not really see a correlation if you're going to look at a really specific food. That's why you have all these levels. So basically, it goes from level 1 to 5, and the first one is really, really broad. So basically, you're looking at fungi, animal, and plant origin of the food. And then you have more detail on level 2. And then on level 3 and 4, you're looking at basically classes of foods. So you're going to see fungi, seafood, poultry, meat, dairy, fruits, vegetables, legumes, seeds. And then on level 5, you're really looking at the specific food. Of course, we're not considering all types of varieties for those, but basically the broader classification. So if you have any type of garlic, that's what's going to populate on level 5. And then just to show what basically you can get from the results, so you're going to get a box plot of those food biomarkers. You can select any of those classifiers. As I mentioned, that's why you need to have the separate table of the metadata to work on the food readouts. And basically you select the type of classifier you want. And then you can select, for example, in this case, we already have many foods to work as an example, but I just selected spinach, arugula, and banana as the numerical information. So basically, it's going to look at the difference on the quantitative difference. That's why also you need to input the quantification table to work on that. And the classifier for this case, as this is as I showed before, this is the example based on the NIST vegan omnivore data set. Basically, the diet information we have for this one is omnivore and vegan, of course. So basically, you can see as a relative intensity the information on the difference of the metabolites related to spinach. In this case, those are the ones that I've selected for the example. So you can see the difference of the intake of spinach, arugula, and banana based on the metabolites that we have curated for those foods. And then also, just going back here, you can select those 3 different significance testing, Kruskal-Wallis, Mann-Whitney, and t-test. And then you can also change some parameters. For this one, you can use log scale, for example, depending on how different it is, just to make it better to visualize. And then, as I mentioned, for all of those apps, since they have been developed in the Streamlit, which is a Python framework for web development, you have the same feature for almost all of them. So in this case, you also have, you can generate the SVG image of these. And also you have the information on the bottom here for the statistical test results, so you can get some hints on how to interpret that data. The second visualization is for a PCA to look at that information. You choose the number of components that you want to look at, and also select the classifier. As I mentioned, in this case, the groups are omnivore and vegans, not going to change for this example. And then you can look at that data, just a regular PCA, of course. Many of us already do that on some script, R, Python, or any other platforms. But the idea is to do that all combined, and as I mentioned, as a streamline of the GNPS job you perform. So we can do all of that almost in a single shot. And the last one is a volcano plot. Of course, you can change that, the thresholds, the groups, that if you have more groups on your study, you can also change that and see what are the most relevant features. It's going to also label for the most significant ones. It's going to label what food they are related to, and then of course the groups that are most present. Any doubts on that? As I mentioned, it's a lot of detail that you can go to look at that data. It's just an idea for you to be aware of those tools, to try that. Of course, you can send us questions later and to discuss that, to suggest other implementations, other ideas that you have. So it's really just, as I mentioned, an overview for you to be familiar with those tools, what can be done with that, and to, of course, to collaborate with your own data, to make it public and to use it as part of the repository idea. But if you're just using the web version, it doesn't mean it's going to be public. That's not what I'm trying to say. Is that the more information you can provide for public repositories, the more information we're going to have to implement on those tools. And of course, more discovery, fortunately, based on that. So the third one is the CMMC dashboard. But before talking about the dashboard, so the tool itself, I just want to talk a little bit on what is the CMMC, which is the Collaborative Microbial Metabolite Center, which is part of UC San Diego and UC Riverside, from Professor Peter Dorstein and Professor Daniel Petrus. And the goal of the CMMC, for the ones that are not familiar with it, is to build microbial metabolite knowledge base. So basically, to gather more information as possible on microbial data, and to do any sort of studies for gut microbiome for microbial cultures, to look at the metabolites, to look at the correlation, to look at information between gut microbiome and other types of studies. And the center is supported by the NIH Microbiome Diet Host Interactions Management Center. And the idea, as I mentioned before, for the drug and foods, we've done a curation of a spectral database for our own samples, for public repositories. It depends a little bit on each of those tools. And for the CMMC knowledge base, the labs that are part of, but any of you that work with microbial data sets can also upload your data for the CMMC. You can just get in contact with them and tell them what type of studies you perform. And then you can also make your data part of the repository. But the workflow for that was basically the deposition workflow, which was basically a data selection of over 30 different researchers that work with microbial data sets. So they selected their data with the metabolite identification. They've performed the taxonomy and phylogeny to provide the metadata for these samples. Also biosynthesis activity, gathered all the reference on papers for that information. They added also grant information and other comments that they have on that samples. So as I mentioned, it's a community effort. It's a collaborative work, as the name suggests. And the knowledge base creation was based on 80,000 spectra, almost 5,000 compounds, 2,000 microbes, with, as I said, over 30 contributors, basically 30 different researchers, 30 groups, of course, that are part of the CMMC. And the workflow for the data itself was basically perform classical molecular networking, feature-based molecular networking for all of these data. And then you can provide CMMC enrichment, and that's basically what you're going to do to your own data too, as we're going to see in a bit. And then you're going to have a rich network. So you have as I mentioned to our colleague before, you have that information on your fragmentation pattern, on the metadata that you already know for your study, but then you do the enrichment. So basically you add more information based on the matches that you have for the CMMC knowledge base. So basically you add that information that it might be, or for some cases you already are certain of that it's related to some strain of microbes or to some host or microbiome study and all that information. And finally, as we're going to see more information now, we have the CMMC dashboard. So basically, the dashboard is how to visualize that in an easier way. For the CMMC dashboard, you have the enrichment task ID. So it's a little bit different, but the ID is the same. So you have your GNPS job. And then, as I mentioned, at the end, if you go to the bottom of it, you have the downstream analysis options. And then you select the CMMC knowledge base. And then you're going to have a second ID, which is the enrichment ID. So basically, it's the new version of your job with that information added. So that's the ID you're going to have on the first page here. And then the second one is the original one for the job you have. So it's the FBmn task ID. And then you can select other metadata that you have that information, just combine all of these together. You're going to visualize the box plot as we've seen before based on the type of information you provided. And then, as I mentioned, the curation of all these microbial information is going to be highlighted on the structure information for those annotations. So you're going to have at least the name, the origin, the type of source, the microbes that it's already been analyzed or detected or studied, and the type of activity information we have on that. And then finally, you're going to see an overview on that, so the visualization of that on your data. And then you can also do a microbe mass integration. As I mentioned before, all the MAST tools are based on the creation of spectral databases, which is ideally, if I'm not mistaken, 4 of them right now, which is the GNPS itself, the metabolites, workbench, the Norman, and there's also another one right now. And the micromass, you can see that information, and also it's going to be released shortly, the microbiome mass, which is another tool for monocultures. So right now we have that's the overview of what we have on the knowledge base. So basically, you have 4,000 microbial molecules, almost 2,000 drugs, diets, information, host, exposure information. And some of those are still ambiguous, that we don't have enough information to be classified on something more specific. And also you have drug information, de novo biosynthesis, diets, hosts, microbial metabolism of food, microbial metabolism related to the host, and also to drugs. As I mentioned, it's not exactly the same information you get for drugs or for foods that if you're using the readouts, because these ones, the information combined to microbial information. So you're not going to have the same information as the other two, and also it's not necessarily the same annotation that you're going to get. So yeah, this is the overview of the tool if you want to access it. So as I mentioned, for the example data, you need to see, and this is already on the website for the ones that are that haven't accessed it yet. I mention it as examples, but that's exactly what you can do on the website. And for the CMMC, as I mentioned, you need to have the FBmn, so the regular molecular network job, but then you also have you need to have the enrichment ID, so that's the CMMC job. You run the analysis, and that's the type of information you're going to visualize on that. So the first one, which is the default, is the data explorer. You can do, you can change the grouping, the statistical test, as I mentioned, for the other tools. But the difference here is that you're going to, on the last step, you're going to select the features that have been annotated to your data set. And then you're going to visualize the information that it's provided for each of these molecules. So you're going to see a comparison test for the abundance, so the peak area information. You're going to have the details for the microbial information, as I mentioned before. So for example, this one, you're going to have the input name, the molecule origin, the source, and then the microbe name, if it is available. As I mentioned, most of those tools, the library, it's always been increasing. So we are always implementing more information, more spectral, and more MSMS2 spectra to that. So they are always increasing. So we might get more results if you do a reanalysis from time to time. And also you get the upset plot with the source, which in this case you're going to see the host diet and microbial correlations, or the origin, which in this case you're going to have the host microbial metabolism of food, some that are still undefined or unknown, so we don't have enough information of that. Then you have microbial metabolism of host-derived molecules, diet, and de novo biosynthesis. Just going back here, the other option, which is if you want to switch a little bit more the parameters, you can go to advanced visualizations. And for my colleague here that asked more about the molecular networks, that's the type of information you get on a molecular network. But in this case, it's showing the specific one for the feature that you've selected for looking at it. So you're going to see the specific network for this feature on the CMMC dashboard. So you don't need to scroll over all the network of your whole job if you don't want to. So you can look directly for the ones that have either the feature ID or the precursor MZ that you're looking for. And as I mentioned, if you want to run a microbe mass, and then the difference that for that, you're going to search your spectra for the whole repository based on mass. So you select the feature that you have interest in, and then you select the parameters on how strict you want that spectra on the repository to match your own analysis. So then you just select the MZ tolerance, the fragment tolerance, the cosine similarity, the minimum matched peaks, and the delta mass that you're looking for. Okay. Questions about the CMMC dashboard? Nope. Okay. And the last one, this one I'm not going to discuss the details, because for that you need not only the information about how GNPS2 works, but also the statistical background. So I'm not going to discuss the theory behind all the statistical tests, but just for the ones that are not familiar, especially the ones that are starting on metabolomics, it's a really good guide, because it not only provides many of those, the most common tools, even for modeling, like some supervised and unsupervised tools, but it also provides a guide as a written guide along with the tool. So you can access the links here, and then you can actually read not only how to use the tool, which is the FBmn-stats, but also what are those tools, what are those methods and those statistical tests doing with your data. So what exactly you need or want to look at while doing this analysis. And for that you have, for the FBmn-stats, you have all these testings here. So we have PCA, Permanovas, hierarchical clustering, heat maps, random forests, and all the parameters that you can select for those. But just an example, if you want to try a little bit with it and maybe ask me something later, you have more information on that. So you can use directly the quantification table and the metadata files if you want to just upload your own data and not having the GNPS job as we've done for the other ones. You can use the task ID as we've done for the drug and food readouts and the CMMC dashboard. You can use the examples from the publication of these two. You can use a small data set, which is lighter and faster to process, just to look at all the possibilities that you can work with. And also for this one, you can use the classical molecular network, and so you don't have all the annotation. Maybe you don't have or you don't need it right now, or if you're working with a really large data set and you perform just a classical molecular networking. For this one, since we're just mainly looking for the statistics, not exactly the chemical and biological information related to the spectra, you can perform that with the FBmn-stats. So basically you select which one do you need. Then you have some information if you want to remove blank features, the type of normalization you want to use. If you want to do some imputation for the ones that are more familiar with statistical analysis. Then you submit, and then all those tests here are going to be available for you to look at the results, to download the tables, and also download the bar charts of that information. And now I'm going to hand over to my colleague, Luís, which is going to present the ChemWalker workflow. And we might get more questions about this one since it wasn't on the initial abstract. So thank you.
Hello, good morning everyone. My name is Luís Feitosa. I'm a postdoctoral researcher at the University of São Paulo, Brazil. Now I'm working at Computational Chemical Biology Laboratory with Professor Ricardo, and today I will present a hands-on of the ChemWalker workflow. We observed that the most nodes in spectral similarity networks are not annotated, and the big challenge is how to predict analogs in the clusters. Thinking about that, Professor Ricardo and collaborators initially developed the NAP2 networking annotation propagation that combines the GNPS annotations in seed nodes with in silico fragmentations by the metfrag tool, and resulting in structural predictions in nodes directly connected to the heat, as I'm showing here. In blue, spectral match with GNPS libraries, annotations of compounds, and yellow predicted candidates by the NAP2, candidates directly connected to the heat. Over the years, the group of Professor Ricardo and collaborators also developed the ChemWalker tool that combines spectral similarity of components of the cluster with the structural similarity of the possible candidates using the random walks in the graph from the seed node, random walks from the annotation of GNPS to predict another nodes, resulting in predictions in nodes not directly connected to the seed node, and therefore increasing the number of predicted structures in the clusters. But how to select the structural candidates? Firstly, you must prioritize structures similar to annotated compounds. And second, you must perform surveys of chemical data in taxa and answer the question, is the occurrence of the metabolite is possible in this organism? You can construct in-house libraries of compounds found in genus and species of interest for the obtain of a list of possible compounds that occur in the organism that you are working. In third, you must perform the manual inspection of the MS-MS spectra, observing fragmentation patterns of natural products, for example, if electrospray ionization. In this case, you must have some experience in MS-MS data to identify the gas phase reaction mechanisms of fragmentation mechanisms, and identify some patterns of fragmentation, neutral losses of some classes of natural products, as well diagnostic ions for some classes of natural products. It's increase the confidence in the annotations. Let's start. You can access a finished job of a feature-based molecular network in GNPS2. After login, you can access the tasks to access a finished, click on the tasks to access a finished job in the feature-based molecular networking. Then you can select your finished job and click on View Task in blue to visualize the page results of your network. Here, an example of the network of actinobacteria that I'm working is approach, this hands-on is an approach of natural products. In the page results, you can scroll down the page and find in the task result links, you can find the network visualizations and click on visualize the full network in browser to access your full network. Then you can visualize the full network here. Some options for coloring the nodes and explore the nodes annotated in the clusters. Here we have a selected node in a cluster. This selected node was annotated as rifamycin-S. And here we have the MS-MS spectra of rifamycin-S. The ID of the node corresponding to this annotation. The precursor ion of rifamycin-S and rotation time. But the most important information here is the component index, is the number of the cluster, the number of this component molecular family. In this case, it's number 28. All the nodes of these clusters presented the number 28, right? And this information is very important to the next steps of Ken Walker workflow. In your browser, you must back to the task result links and access the Ken Walker workflow. Please scroll down the page at the downstream analysis and click on Run Ken Walker Analysis to start the workflow. Let's set the parameters in Ken Walker. Firstly, we have a job description. You can insert a name as you choose. In the mandatory parameters, firstly, we have the ID of feature-based molecular networking job imported automatically. Please don't change this ID. This ID is corresponding to your analysis of feature-based molecular networking. In workflow type, I'm working in GNPS2, and consequently, I'm selected the GNPS2. If you are working in GNPS server, GNPS1, you can select the feature-based molecular networking or V2. Here, it's important, as I mentioned before, the number of the component index, in this case 28, to predict the structures in the cluster, exclusively in the cluster 28. Another parameters, ionization modes. I obtained the data in positive mode. Adduct, I'm selected the protonad adduct, but you can select adducts in a list as ammonium adduct, sodium adduct, potassium adduct, for example, and adducts in negative mode. You will see predictions related to adducts. ppm tolerance, you can increase or decrease the tolerance. Default is 15. If you obtain the MS data in equipment with higher resolution analyzers such as TOF or Orbitrap, you don't must increase the tolerance above values, above 10 or 15 or 20, because you must keep the high resolution, and you must choose the predictions that present the exact mass close to the observed mass to keep the high resolution and increase the confidence in the annotations. Click on Submit Workflow to start the analysis and wait a few minutes for the finishing. Here we have the page of results of GNPS2, results from the Ken Walker analysis. In this page, you can download the compressed file if results by clicking on Random Walk Structures GraphML and wait a few seconds for the download. Here, you can extract the compressed file to a folder. Click on the right button on the file, and then clicking in Extract All. Select a folder for the extraction, and then click on Extract. Well, you also have to install plugins for the Cytoscape, 2 plugins. The first plugin is the CanVis2. Here's the link to access the page, but you search by CanVis2 in Google and access the page and download the file. In the same way, we have a second plugin to install, iFiles Layout Algorithms. Here is the page of this plugin, but you search by iFiles Layout Algorithms in Google and access the page of the plugin and download the file. We assume that you have the Cytoscape software installed in your computer. Let's open the Cytoscape software for the visualization of the cluster. After open the software, you must drag and drop the graph.ml file previously extracted, and you will see this cluster, and then you must install the plugin. First, you can access the on main menu apps, select the App Store, and then install apps from file. You must select the folder that you downloaded the plugins. Firstly, you click on Canvas 2, and then open for the installation. In the same way, click on EI files layout and click on open for the installation. These two plugins for a better visualization and organization of the cluster. You can go to App Store tab in left side of the window inside Escape Software and check if the plugins have been installed. You must see the two plugins correctly installed, and the box was checked automatically. Well, you must format the cluster visualization, format the nodes by fill color option, change the color of the visualization, the shape of the nodes, and the size of the nodes. I enlarge for a better visualization of the structures of candidates, and don't forget, check the box look node width and weight for a better visualization. You can take a picture if you want some. Yes. You must organize the cluster using the iFiles plugin. You must click on the main menu, scroll down at the, click on Layout in the main menu, and scroll down at iFiles, Organic Layouts, and we'll see a better organization of the cluster. You also must color the interest nodes, select the color in third column of fill color option. Then you must click on the node and select a color that you want. I painted yellow the node with the candidate I want to see. Let's set the display of structures. You must click on App Store again, select the Cheminformatics tools, and click on Settings. In Set Parameters window, select the node smiles. That is the representation of the predicted structures that you must import for the visualization of structures. You must paint structures on nodes. Click with the right button on the selected nodes. Select Apps, then Cheminformatics Tools, and paint structures on selected nodes. You will see the structures of predicted candidates to all the nodes of cluster. And it's important here, you can observe all candidates to a node. Click on the right button on the node and select Apps again, Cheminformatics Tool, and show structures for a selected node. You will visualize many structures predicted to only one node. And you must select the structures that make sense for your study, make sense for the organism that you are working. I'm selected the structural analog of rifamycins with the naphthoquinone derivative ring, SU chain derivative, known as ANSA chain in rifamycins. And this structural analog are in agreement of the chemosystematics data, since these analogs were produced by the taxon of actinobacteria that I'm working, as I found in literature. Then you can access the CanVis window. Click on the CanVis button in the right side of the window and explore the list of candidates. We will see again all candidates predicted for the yellow nodes. And I'm selected the rifampicin derivative. And here in PubChem link, you can access the PubChem page of this compound to find another information about the metabolite, such as spectral information and NMR spectra, if available, MS spectrum, information about the bioactivity of this compound, and molecular descriptors, and among others, informations. And finally, an example of results from a spotted figure by Cytoscape software, spotted figure of the cluster that I'm showing in this presentation. Rifampicin S corresponding to the orange node annotated by GNPS, and yellow node corresponding to annotation of Ken Walker, analog of rifampicin, and the node was not directly connected to rifampicin, showing the application of Ken Walker workflow. For more information, you can access the documentation of GNPS2, search by Ken Walker workflow. This is GitHub of the codes of the application. This is the paper of information of the Ken Walker. Please meet our group, group of Computational Chemical Biology Laboratory, scanning the QR codes. This is my email. Thank you. Questions? Any question?
Good morning. Great presentation. Congratulations. It may sound silly, but I'd like to know how do you choose your adduct for each cluster? Like, we have the IO identity information from MZMine, for example, or you do it another way. That's I believe it matters for the Ken Walker algorithm, right? The adduct choosing.
Firstly, we have to thinking about the annotation, confirm the annotation in as the 3 steps that I showed in the presentation. First, I confirmed the annotation, and to identify if the structure was protoned or with sodium, potassium, and then I'm selected the candidates that possibly connected to the hit node. For example, a cluster candidates connected to the node. The candidates connected to the node showed if these are protoned. The analog also protoned. If a sodium adduct here, a sodium adduct, it's in another cluster, not here, right? You can observe adducts in other clusters with sodium, potassium, among others. It's, I'm correctly
Yeah, makes sense. Makes
this-
sense. And do you discuss the results with the annotations from Cam Walker as well, with trustability?
Discuss the
Yeah, your results. You use the annotation from Cam Walker.
yes,
I assume.
yes, but we have to confirm the annotations in the third step. We observed the pattern of fragmentation, if this pattern is compatible with the class of compounds, to confirm the annotation. Always, always.
Perfect. Thank you.
Hi, thank you so much. Here. Thank you so much for the presentation. I had a related question because I did try it, K-Walker, I liked it a lot, so thank you very much for that. I had, for example, like one cluster that had proton adducts and ammonia adducts, like everything kind of connected together. So if it would be possible in the future to add a function where you can have more than one type of adduct, or if I should run 2 times K-Walker, one, for example, with proton, one with ammonia, or yeah, how would it work when you have more than one adduct in one same cluster?
Kim Walker is like, has to be developed by a multidisciplinary team, right? He's a chemist, you can see. So he's talking like a chemist. So we need developers to create these algorithms. And our colleagues was right. We could use, for instance, ion identity molecular networking because it will give us different adduct predictions inside the same molecular family, inside the same cluster as you wanted. It's not implemented right now, so when you choose an adduct type, it will try to guess if the accurate mass of each mass peak matches the mass of the adduct that you chose. For now, we cannot, but we want to change. Luís will participate on development of improvements of Chem Walker, and that's one of the first improvements we are going to do. Okay, thank you for your question. Any more questions?
Quite interesting, but this is related to the earlier presentation. So there was this threshold, if I remember the number is 50,000. So would this threshold that you choose for identification of different, potential compounds, would it be dependent on the platform, dependent on the sample, or dependent on the, how you process the sample and so on and so forth, including how you process the data in the MG mine? So is it like there is a thumb rule that you have, or you try and see, trial and error?
Maybe Luís wants to complement, but I mean, we basically are basing it on the mass accuracy of the instrument, So generally, when you have like a recommendation for the all platforms, you usually try to guess based on the mass accuracy of the instrument. If it's a QTOF or it's a Orbitrap, so you set the mass accuracy threshold based on that. That's not mass accuracy, what?
This is related to finding drugs and stuff. Yeah, I'm sorry. So that's where, while I'm trying to fish for, what all drugs are present or what are the food ingredients, compounds present, how do I decide the threshold, peak threshold?
Okay, so it is a similar answer, but you can, initially you do that by trial and error. But for example, for that one, the example, which is kind of a high threshold, it's 50,000. So for this one, for the example, was a really specific case because one of the drugs were actually detected on many samples on a higher threshold than expected. But just because we know that information, we could use that for the example. But usually you're going to set as 0 and see whatever appears. And then you of course, you're going to look at the type of instrument. Usually for the Q-TOFs, you need to set a higher threshold. The Orbitrabs, you can set it lower because of the sensitivity of the instrument. And also, it's going to depend, as you already mentioned, you kind of already answered that, depending on the protocol you used for the extraction. For example, you're probably going to see more of certain kind of drugs than the other ones. And also, the other thing that's going to change a little bit is basically when you actually perform the whole analysis of the features, and then you keep track of for example, the carryover effect. So depend on the amount of carryover you have on your samples, on the blanks and the pooled QCs and things like that. And then you can actually set the threshold. But ideally, you can set to 0 and do the whole screening of all the matches. And then that's one of the reasons I mentioned the restart session for the analysis, because then you can set a different threshold. So for example, for that one, we have the 50,000 because one of the drugs we already knew that was present there. And also because we have one of the internal standards is also a drug. So that's why we could keep track of that information. But when you are doing an untargeted that you don't have that many information on the actual things that you're looking at, basically my suggestion is set as low as possible. For the drug readouts, I think the lowest we allowed for now is I think 100 or 1,000, I'm not sure right now. Just because 0 is going to show everything on that, it's going to be a mess. But basically, I mean, of course, you can do that directly on the job, but just for the visualization, just because it's a little bit lighter on the server, I think it's 100 or 1,000 for the threshold. But basically, you're going to see almost everything, and then you set a higher one, depending on the instrument or specifically the molecular class or something more specific that you want to look at. But it's still, if you're not tracking, as basically the idea is for untargeted studies, if you're not tracking something really in particular, set it as low as possible. And my suggestion is just be strict on the other information. So basically the number of shared peaks or the cosine score that you want to look at. And basically then when you set the drugs that you're really interested in, and then you can basically set an actual threshold for the abundance or the peak area or anything like that. But save that, I mean, be strict on the abundance itself, or basically on the actual statistics for your visualization and interpretation as the last step, not as the first one. So basically look at the annotations, look at the chemical information that you can get from your data, and then when you're actually looking for the biological interpretation, then you be more strict and look for things that actually make more sense on the pathway or on the type of study that you're performing. That's the idea, but you don't have a set threshold for any type of things. That's going to basically be dependent on the platform that you're using, on the actual state of your instrument when you're running that data set.
Now, Professor Ricardo will present the GNPS2 server in Brazil. Thank you.
Good morning, everyone. Who is hungry here? I will not keep you from lunch anymore. I'll just close with, it's an announcement, actually. So, where is it? GNPS2 is becoming a federated infrastructure. It will merge, I now think, 7 institutions in 4 continents. So the idea here is that we will have these servers in different locations, and the aim is to decrease latency, so you will be able to exchange data fast, and also distribute compute. So if you are from South America especially, or Latin America maybe, you probably want to link your account to the Brazilian server. So we are hosting this Brazilian server at School of Pharmaceutical Sciences at Ribeirão Preto in Brazil. So you don't need to do this now. I think most people are taking pictures, but we also are going to share this presentation so you can follow all the links, all the explanations we perform here today. And this is very interesting for us because it gives us the ability also to know the development of the platform closer. So if any of you have an idea for a tool or an app, all this code is available on GitHub of the main developer, that is Ming Wang at Riverside University, as Lurian mentioned. So you can go there and see how these workflows are being developed. So if you and your group of collaborators have your own idea for a workflow, you are welcome to try it and maybe connect to Ming to contribute. So that's basically it. After you do this connection, this password management tool, you'll be able to access the server in Brazil. We now have only one workflow of feature-based molecular networking, but if you are kind enough to adopt this server, we will add more workflows. With that, I just wanted to thank Lurian. He did a great job preparing this workshop, and he was kind enough to accept us to come here and talk a little bit of ChemWalker, and now to do this advertisement of the mirror we have in Brazil now. So if you have additional questions, you can ask now in the last few minutes, or you can contact us later. Thank you very much for coming. Você quer colocar o link agora?
So if I can still have a question on ChemWalker.
Sure, yeah.
I was wondering whether you had a chance to compare it to some other in silico tools, like Sirius, for instance.
We did not directly compare with other tools. The only, both for NAP and for ChemWalker, the idea was not, we are not doing the in silico fragmentation from scratch. We are using in silico fragmentation predictions given by other tools and trying to propagate the information from spectral library matches on the networks and improve the ranking of in silico fragmentation tools. ChemWalker is not a different fragmentation tool itself. It just merges the data you observed on spectral library matching with the in silico fragmentation. That's why we haven't compared. But we have a lot of literature now. If you go to Google Scholar, ChemWalker still has a few citations. NAP has like over 400 citations. You can find a lot of articles where people combined it with Sirius, and Sirius is like, through CSI Finger ID, is the best performing in silico fragmentation tool we have available. But sometimes it misses some annotations, and that's because most in silico fragmentation tools, they perform annotation on each node or in each peak individually. So the idea of ChemWalker or NAP is to combine all the information in your network and come up with like a set of predictions that make sense together. So there is a lot of literature showing that when you combine these tools, there are some cases where the best performing tools like Sirius misses some of the annotations. I don't know if I answered you.
Yep. Thank you.
Yeah. I can, if you don't have any questions, that was it. But if you have questions, feel free to stay with us and talk a little bit more. I think I'm just going to create a QR code for this whole presentation here, if you want to have it. If not, we can also email you later. We will ask the organization for the emails of the ones that were subscribed to this workshop